Is everybody able to see the screen? All right. So here's an overview of the high level structures in the team and organization. I already kind of I went into some of the discussion about the structures just now because I didn't have a slide for it, and wanted to make sure that it was addressed in the discussion here. So again, we're a scientific NSF institute that is built on the strength of 6 institutions with many different departments and investigators. Several grad students and postdocs are involved at various levels of this organization. We have several postdocs that are assigned to the project as well that we're funding from external sources that we're able to use the institute to unlock so that we can add more to the team to try to bring more resources to bear to accomplish the scientific mission at which we're working on. So I'm the PIN director. Worked closely with a lot of the thrust leads on capturing the vision of what we wanted to do together, along with Chris Tiger, associate director or deputy director, depending on which email signature is coming out. And Anjali Iyer Pascuzzi, Jeremy Zartman, Janice Evans, and Brent Ladd, as well as Alejandra Magana, and Stephanie Gardner has stepped back due to a nice new role in her college, but still very actively involved in Thrust 4. Here's a very high level overview of the horizontal and vertical structure of the Institute. So we're organized vertically by different biological thrust areas, as well as with an overarching thrust 4 to take what we're learning across the Institute and incorporate it into our roles as instructors and teachers in the undergraduate and graduate schools space. At our core, we're really looking at a small number of things in these biological systems. We're looking at calcium sensors, of which there has been a lot of data coming out that requires a lot of annotation and careful processing and sorting from in the data science space, as well as increasing amounts of actin information that's coming out throughout the Institute. So to capture that information, it relies largely on imaging using a spinning disc confocal microscopes, and some light sheet microscopes, as well as some AFM, but it's very much in the light microscopy family, fluorescent microscopy family. So there is a lot of consistency in the data that is being generated because the reagents are producing similar signals that are picked up by similar detectors, and these detectors in these sensors work across institutions. These sensors work at single cell, multi-cell, and organism scales. But what's unique about the Institute and where we're encountering a lot of challenges as part of our integration is the while we're measuring similar reagents in multiple biological systems, there's a big part of our institute that's based on image processing, image analysis, coding, as well as simulation, core simulation integration. So this is where we have growing expertise and a team that Nilay Kumar brings over, that Lin is now able to devote to the Institute 100% of her time, that Sharon Minsuk is bringing, and also with Adrian, we're having a mechanical engineer from MIT join in 2 months following his PhD defense, on Lillian Gilbreth, who will be focusing a lot on the simulation integration as well. Like I said, there's ongoing work in identifying some additional personnel support to support a lot of the core data integration. Hopefully have more on that very soon. So this is the horizontal integration, where we take what we're measuring and we're trying to capture what we're measuring with very reproducible quantitative ways. And we want to bridge the vertical scale through simulation and integration that we're getting from the image analysis. This is the the focus of the work and the challenge all in a figure. The way that this is organized is with the biological systems across these different scales, starting in the high nanometer scale, all the way up through things that approach, the small sub-centimeter scale. So we're looking at spatial scales that span 6 orders of magnitude in terms of the overall size of the organization. The concept is to measure the same sensors and variables in these different scales, and then use shared frameworks for processing these so that we can have consistency in how the data is viewed and utilized. This is really a picture that summarizes what an institute could accomplish in 10 plus years, as we're encountering that the process, the progress has been really actually quite substantial in many of these, and we should start to highlight, where we have signaling measurements in these different things we're looking at measuring in these bubbles here, and then start focusing on where we have the gaps. But we are getting this data is coming from all different aspects of the institute, and this is data that didn't exist, a couple of years ago. The challenge and the opportunity is in how to use models to bridge these systems. So it's going to be it's really interesting to think about the core concept of these being shared pathways with shared primary components, that if you were to knock one out and you have a phenotype, you could take the equivalent from another one of the biological systems, knock it in, and expect rescue. So with that kind of underlying principle, you don't expect there to be dramatic differences in the function of the protein that you're swapping back in. And so there is this core of the regulatory networks that we're seeking to untangle with this approach. And that core of the mechanism, what sits in the mathematical models and bridges the input data that is being acquired across the institute with the expected measurements and outputs, which are simulation results. So this is a nice figure inspired, looked at, excuse me, from some work that Jeremy's done to show how multiple types of inputs lead to modifications of some shared intracellular processes. There's feedback on these intracellular processes, but ultimately the outcome is looking at how these impact cytoskeletal dynamics and coordinated remodeling. So the ones that we're focused on in our institute are mechanosensing and chemosensing, things that are triggered when you have an injury or a pathogen attack. And then there has to, and the output is a cytoskeletal response that prepares the cell to survive and be robust against these perturbations or these input stresses. This is where the term robustness comes into play, as opposed to the quality of the flavor of a fine wine, for instance. The robustness here means the ability of the system to overcome, these types of stresses and to do so reproducibly. Here's some examples of data that's been collected from Jeremy's group and from Ching's group and from Chris's group. And this is simulation from what El Chupanar worked on with some input CompuCell 3D from James, to look so you can see this is one of the sensors. This is actually old data from last spring. There's more now that's more up to date, looking at calcium sensors under various conditions in Drosophila, in plant cotyledons, and in zebrafish tails. So the institute organization is built across scales in terms of spatial scales, but also to some extent in terms of the total number of cells, the complexity brought on by the total number of cells. The basic building block of life is a cell. Cells are usually within a relatively small window of actual physical size, and you just get bigger by having more cells. And so that the organization takes a jump from single cell to multiple integrated cells that are discrete to large groups of cells that are really hard to manage and think about as discrete individual units. Throughout all of this, though, there's studies going on about how the Institute functions, about how to do integration training, about how to develop new materials so that the tools that help bridge skills, which largely come from simulation and mathematical modeling, are accessible and useful and can be developed by folks with different backgrounds and vice versa. This goes back way back to, if you talk to two different people that are starting a class, one's going down mechanical engineering route and one's going down biology route, and you ask them to draw a vector. One of them's going to draw a circle with some A's and G's and T's and C's, and the other one's going to give you a line of numbers arranged in a column. So there's differences in language, there's differences in backgrounds that we bring to these problems that we work on together. And part of the goal of the Institute is to study how to do that as we're doing it. So there is some building of the airplane while we're flying it. And that is the case for all of these BIIs. And so Stephanie is doing a lot of work in that space, Dr. Gardner, to understand how that happens and how that works, as well as Alejandra Magana and Alsha Pinar in the classroom setting. So a lot of interesting pieces to this puzzle. So there are some examples of how the research is integrative. And these were prompts that NSF was asking before about the different types of integration you could have. One of the examples is bridging. This is the applying principles, findings, and models from one species and applying it to the other. So this is an example where Chris and Jeremy's groups have been working together to bridge a lot of what's been working over in Drosophila into the plant into the plant system. This came out. It's supposed to have videos behind, but that's kind of the general approach that's taking place at that level. It also is informing Linlin and others on some of the ways that we have to think about, including doing this type of modeling of calcium in the enveloping layer in zebrafish. And we just had a study this afternoon that was unpacking a paper that's similar to what Sharon Minsuk is working on in terms of looking at vertices of cell positions in 3D for an overall embryo, but then how that relates to the mechanics of what's happening, as well as where vertices would be to define an epithelial sheet for a cell that is part of this epithelial sheet wrapping around the embryo. And ultimately, the link there, takes the center points, centers of mass, and then is able to identify hypothetically where these vertices would be by drawing triangles between all these centers of mass and these vertices. You need to make triangles to develop a finite element model. You need a finite element model to do the calcium type of wave propagation and signaling. And so there may be a way that kind of technology can be used to bridge the modeling types that are needed to be modeled together, which are a little bit of a challenge right now. In addition to the biological aspects, there are challenges of integration of different types of modeling that we're encountering regularly as a modeling team as well. Other types of things that we're working on, there was a concerted effort from work that was led from Norma, who is now doing a postdoc with Genelia Farms, and Maurizio on looking at the best practices for calcium signaling in a journal, calcium signaling quantification in a journal club, and identifying that there is a need with Daver in Maurizio's lab taking a large role in looking at developing a way to do post-processing that could be consistent and shared between different systems for the Institute. And so where we're at is we still need a lot of work on data inventory, on how do I go back? On getting the data archived in a way that can be continue to be accessed, processed, and evaluated from those that are developing the simulations and then feeding back and forth, but also accessible as we want to start bridging between the different thrust scales and sizes. So going forward, we want to continue. I just want to remind everybody that, structurally, we're going to continue to have these meetings. We're going to focus on the science. We'll have additional journal clubs. We'll have the newsletter that continues to go out, and the PIs meeting, and then the summer workshops. But we have this new resource that we're going to really prop up and work with, not to overwhelm them, but to help be the glue, help provide some advising and some glue and support and communication with some of the research scientists in the Institute that are going to do this in that important role. So I'm going to stop my sharing right now so we can have some more time for Q&A and just general discussion about things that you're looking forward to, that you may, having some different thoughts on, and next steps that you can take individually within your thrust area, or think about ways to bridge across those. I've got a mixture of PIs and students here today, and so we can take questions from anyone, but we can ask students first. Want to pick on individuals, but know that I got some really cool pictures and texts from Juana Mendenhall was sharing some of the outputs that Jasmine was doing with the reagents that Jeremy sent to do some testing on the AFM last week. But I think that can just open it up for all folks for questions and discussion. That means either everybody was completely lost in the presentation.
I'll go ahead, because I was fortunate enough to have heard you commenting about Juana having texted you this bit of information about some exciting things that Jasmine was doing. And if I remember correctly, some fabricated objects that are mimicking the wing disc.
Jeremy sent wing disc, actually.
Actual wing. Oh, that's right. It was fixed wing.
After the PI's meeting.
We're gonna start to image those. So we've been, actually, it's an undergraduate student that's doing the printing piece. Yeah, Xavier. And between the two, they're working on the printing piece and the AFM work. So we have a few things in the pipeline, and I shared this with David, I think, when I was there. The Berkeley National Lab actually has a AFM with an IR attached to it. So I'm writing a user proposal with one of the scientists there to actually have Jasmine go get trained on it, so we can actually image AFM and IR in real time using the tool at Berkeley National Lab. So we're moving in the right direction. It's just a process to get there. But things are looking good. So yeah, I'm excited.
And any good lessons learned from trying this on the, is the purpose of doing it with the fixed wing disc material, just to figure out like the scale of the probe you want to use?
And things like that. So that's what we're working on. I got a checklist for all of that stuff, the materials we're using. So now we've essentially established like the protocol, the printing conditions for a particular material, but then we'll start to tweak some of that when we learn about the mechanics. So to have the materials that are comparable to the mechanical properties. So that's what we're working on. So again, moving in the right direction. It's just a process to get there, but I see a little glimmer of light at the end of the tunnel. A
Cool.
glimmer.
Yeah, hey, we take all the glimmers.
That's it. That's
Yeah,
it. But
and
you
so
got to stick with it.
were
Yeah,
you
so it's
finding
really
fixed
nice.
material worth working with?
So it's interesting. My undergraduate student had a idea for a different method, and we're working on something really cool to kind of market EMBRIO. So I shared that with David as well, but we're doing something even more cool with it. But the method we attempted to do didn't work out that well. So we're tweaking it a little bit. It was something called the fresh method, where you can kind of print. It may work better for Mary's work. So anyway, with the cell, with the oocytes and even the epiboles, right? So that's what we're kind of learning. The fresh method, how Mary, may work better for the cellular work versus things such as the zebrafish tail and the wing
Right,
disc.
the complex tissues.
Yeah,
right,
yeah. So but yeah, we're I'm excited about it actually. So
Well, and
it's
as you
coming
might
along.
guess, I'm asking, just thinking about, how could this be applicable on our end? So maybe
No.
I'll
Exactly.
follow up
And I think
separately.
that's exactly where we're moving with that. And so that was a lesson learned, because we tried it with the wing disc, it didn't work well. The wing disc print actually worked better without this support system that we thought we needed. So but I think it will be more ideal for the cellular prints. And we've kind of assessed the dimensions of the wells that we have to print in, and working through the extraction piece to actually get the cell mold out of that and things like that. So we're working on that end and figuring out ways to actually get to the actual organism or cell that we want to work with. So processes along the way, lessons learned, and trying to be able to tell the story at the end of the year. So it's exciting, but I got to micromanage it a lot. Unfortunately, you guys know this, but I have undergrad, so it's you roll up your sleeves and get in there so you can get
right there.
Get it done.
Yeah. Well, good on you. Thank you.
Mm-hmm.
It's quiet relative to the lively discussion at the PI's meeting, but I get it. It's kind of, as a mixed group, we're getting into some of the troubleshooting and ideas of next steps. I totally get that. Jeremy.
Yeah, actually, just one thing, during Journal Club last week, there was a question about Slack channel in terms of older messages are no longer accessible because it's past the 90 days for the free accounts. So I guess one question is whether Slack could be better utilized for keeping collaborations active and through pinging each other with messages and communication, or if there are better ideas out there.
That's a good question. I think the primary conduit has moved to that newsletter because it was the common denominator, but it doesn't always necessarily get taken up. How effective has Slack been in general?
I think it's perhaps the easiest way to do one-to-many communication set, especially if you're on like a journal club or other sort of.
How about pulling the postdocs and students? Are you still actively engaged in the Slack?
We have a
general?
user that is used in general clubs, and I just created one for the scientists to be able to communicate it between us. I think it's good for the group instead of like an individual message. Sometimes it's not popping up very kind of efficiently. You miss that, but I think it works for when you need to send it, like having a group chatting or like between the groups. So we have been utilize that one for the general club and in small groups.
Yeah, I think we've hit some limits with the free account that we have. So in order to make it functional going forward and adding people that are coming into EMBRIO that are new, we probably have to look at a subscription. So we'll have to look in to see what that'll, how much that is. But if it's useful for enough people, then it's probably worthwhile.
Mm-hmm. as some of the other coding things to, develop in the way that the data is going to be, with the data, director kind of manager position, there might be some movement to other types of, platforms as well, GitHub and others, which are better for coding. We'll have to see how that integrates, into this. Hayden, could you provide just a high-level update about some of what was discussed recently in regards to what you're working on for the codifying the computing training materials and the development that you were doing?
Yeah, so for those who aren't familiar, we've been working for the past couple of months on trying to put together a online ebook style training course on multi-scale modeling in general, but also on using CompuCell as a platform for doing some of that. We've also been talking about developing some tools for Tissue Forge. We've been working on getting a prototype of the site up and running. I've been doing a lot of feature testing over the last couple of weeks to make sure that the different combination of packages that we're trying to put together will work for everything that we need. I think we've got that pretty much settled. We've just been starting to drop in content. So yeah, I'm excited over the next couple of months to work with various people in the group about determining how to best customize that material for kind of what different arms of the organization need from that. Yeah, James is here. The one issue we've run into is we have so we're using a platform called Sphinx to help generate the documentation for the textbook. One of the reasons for that is so that if other people want to start helping, it's actually a very easy platform to learn. I've been putting together some documentation for that as well to get people up to speed for anybody who may want to help with adding content to the various pages once we have the site go live. The one issue we are running into, sorry, somebody just started an air compressor right outside my window. Hopefully that's not audible through the microphone. But we are able just to pull in Jupyter notebooks, more or less whole cloth. So if there's Jupyter notebooks that exist, we can kind of pull them in as individual pages in the textbook. The only issue is that it gets rendered in as static HTML. So it just basically shows all the contents of your Jupyter notebook, but you don't have the ability to work within live cells inside of the page once it's rendered. There's a couple of workarounds that I've found for it, but they're all a little bit clumsy. So we're still trying to kind of work on that piece, but if anybody has any leads on potentially how to just easily embed live Jupyter cells into HTML, that would be useful, because otherwise I've got a couple other branches to investigate, but yeah, so that's kind of where things are there. There will be by the end of this month, we'll have David, you and I will probably need to talk about how we want to do hosting for that at some point. I don't know if there's any particular methods that EMBRIO likes to use for hosting content like this. But once we get that figured out, we can get the prototype live and start getting feedback from people.
That'll be a very useful resource for a lot of trainees. So looking forward to that. There's been so much developed over the past years. That would be great. Well, we're on time, folks. We will send out the updates soon for the progress reporting due in the beginning of April, and more to share soon, as well as the updated schedule for the spring term for these meetings as we start to move around to different thrust areas, report out a lot of the cool science that was shared at the PI's retreat. So look forward to all that. Thank you, everyone.